Okay, so we got some practice on chi-squares, and we're now masters once again on that. And we're looking at Thomas Morgan and his examination of fruit flies. And what's happening is they're looking at two different traits, body color and wing shape, simultaneously. So we're doing a dihybrid comparison and looking at the possible outcomes of two flies or two types of flies which have been crossed. Now the first type is heterozygous for both body color and for wing shape. So they demonstrate the wild type gray body and the wild type wing, and that's being crossed against a homozygous recessive for the recessive trait, which is black body and vestigial wings, which are little winglets that aren't really functional. And when we set up a chi-square for this, when we set up a dihybrid cross for this, we see that there are four possible outcomes for what the different phenotypes and genotypes of the offspring could possibly be. So going into this experiment, there's a expectation that there'd be a 1/4, 1/4, 1/4, 1/4 ratio of the possible outcomes. Therefore, when they're looking at 2,300 flies, that's going to be 575 flies expected of each of the four different types. However, the observed results were wildly different from that, and when we run a chi-square, we see that we pretty emphatically reject the null hypothesis, that we are very, very, very significant in the difference between the expected and observed results. So the question then becomes, well, what's going on here? Because Mendel showed us pretty clearly that this is a tool which we can use to understand the independent assortment of genes which occurs as a result of meiosis. Now, pretty quickly, the idea which was identified is that these genes, for some reason, are being carried together. And this is the early days of understanding this, but what Morgan's showing here is the fact that the gray versus black body gene and the vestigial versus normal wing gene is carried on the same chromosome. The significance there is that, this is 40 years before Watson and Crick. This is 40 years before Hershey and Chase even definitively showed that DNA was the mechanism by which genetics are passed on. But the point is that here in 1909, they're demonstrating a deeper understanding of how these things actually get passed on, that it's not just a mess of genes and randomly they all sort out independent of each other, but refining Mendel's work to say that, yeah, that's reasonable and that's a good expectation in instances where we don't have a relationship between these genes. But in this case, we clearly do. The next problem, though, is that if the body color and wing shape genes are contained on the same chromosome, then there would be an expectation that the recessive one is going to pass on a little b and a little v no matter what, right? But the other fly is capable of passing on a big B along with a big VG, or a small b with a small VG. And therefore the expectation was that, okay, if these are on the same chromosome, the same thing that's actually being passed forward, then we should only see either gray body and normal wings because they inherited both the large B and the large VG, or we should see the other half should be the black body and vestigial wings because it passed on the small b and the small VG. But of course, this still is a problem because we do have 206 gray body with vestigial wings and 185 black body with normal wings, and these shouldn't be together. There shouldn't be a possibility for these to even have been put forward because they would have been on separate chromosomes. And so this is the early understanding of recombination, or specifically the crossing over event occurring during meiosis, where we can actually mix up which genes are being put forward, that even though this organism had one chromosome that had the big B and the big VG, and one chromosome that had the small b and the small VG, it has somehow passed on a flipped version of that where it can pass on a mix of this, but not always. And so this identification of certain cases of recombination then led them down a new path of trying to understand, okay, well, what's the likelihood of recombination? And if these are physically located on a chromosome, are there characteristics, or is there a reason why certain genes might be more likely to recombine than others? And so as we continue through this and we find that, okay, yes, we have pretty clearly a reject the null hypothesis in favor of the alternative hypothesis. This told Morgan everything we just went over. But now we're looking at, okay, so what is the frequency with which this recombination is occurring? And if we do this, we find that the recombination frequency is 0.17. 17% chance of recombination occurring, and that's because we have 206 plus 185 out of the total 2,300 flies in which this occurred. So 17% of them involved recombination of some sort. The next part of this activity asks to take a look at sort of a hypothetical situation. If we're comparing three different genes, gene A, B, and C, can we use this information about recombination to tell us a little bit more about the actual physical location of these traits on the chromosome, that by looking at the likelihood of recombination, can we learn something about how these things are actually situated within the chromosome? And it turns out we can. And so for this first one here with number 7, we're looking at a comparison between A and B, and we do the same test cross that Thomas Morgan had done with the flies. We see a heterozygous individual for both traits crossed with a homozygous recessive individual for both traits. In this case, the parental genotypes are the big A, big B, little b, and little. The offspring that are recombinant offspring are going to be the big A, little b, and the little a, big B, little b. So the point there being that these two genotypes shouldn't be possible except if recombination has occurred. And so to find the recombinant frequency here, we look at how many were recombinant. So that's the 50 plus 50. So that's 100 out of 1,000, or 0.10. So about 10% are recombinant. So through this, we come up with a way of mapping these genes. And so what we do is we take that number, multiply it by 100, or basically turn it into a percentage, and we call that a map unit, which is in units of centimorgans, named for Thomas Morgan. And so we find that the A and the B gene are 10 centimorgans away from each other. Now, if you're wondering, like, well, how far is that? It doesn't really equate to a specific distance. So it's not like saying nanometers or centimeters or anything like that. It would be on the scale of nanometers. But the reason why it's not a precise distance is because there's more factors involved here. It's not just strictly how far apart they are. Yes, that's a meaningful thing, but it's meaningful because, as we see with Barbara McClintock and tons of researchers beyond that, there are certain factors which influence the possibility of crossing over, things like transposons and these specific segments of chromosomes that are more likely, because of a whole bunch of chemistry and a whole bunch of proteins, more likely to be involved in the actual flipping. So 10 centimorgans is a way of understanding, in a comparative sense, how far apart these things are. But just looking at A and B isn't enough. And so then we bring in gene C. And now we're going to compare A and C. We're going to run another dihybrid. We find that there's 460, 460, 40, and 40. Again, the parental genotypes are the heterozygous for both and the homozygous recessive for both. And in this case, our recombinant ones are basically the other two. And again, we do the same thing. So how many, what's the recombinant frequency? It's going to be 80 out of 1,000, which is going to be 0.80, or 8 centimorgans is the distance between A and C. Again, standalone, that's not a great piece of information. All that tells us is that genes A and C happen to be a little bit closer together than A and B were. But in terms of mapping these things, we don't really know much about the actual location in a larger sense. So we do the third comparison. Now we compare genes B and C, and we do the same thing. Okay, the parental ones are going to be the heterozygous for both, and the homozygous recessive for both. Our recombinant ones are going to be the other two, which again would only occur in the event of crossing over. And in this case, we see actually much fewer recombinant instances, and so we have 20 out of 1,000, or 0.02. So we know that these two genes, B and C, are 2 centimorgans apart. Okay, now we can take all 3 of those comparisons and build a bit more of a map. So I know that there is a distance of 10 centimorgans between A and B, but I know that there's only 8 centimorgans between A and C, and 2 between C and B. And so using that information together, we can find out that A is going to have a distance of 10 centimorgans to letter B. And if I put C here in the middle, and I know it's a little bit hard to see this, but basically looking at these 3 blanks, it goes A, C, B. And if we compare the centimorgans between these, we'll see that it checks out that because the distance between A and B is 10, that tracks because the distance from A to C is 8, and the distance from C to B is 2, so it's a total of 10. And now what we found pretty definitively is that gene C must be located between A and B, and we know that it's much closer to B because there's a much lower likelihood of B and C separating from each other. Now, it's not to say that there's not a high likelihood that crossing over is happening. It's just that if crossing over happens in such a way that both B and C are together in the segment that gets crossed over, then we see those stay together, which is why we don't see much recombination when it comes to B and C, because they happen to be very close. So the only way in which they could be recombinant is if the karate chop of crossing over happened in the small space between them, which obviously it can happen. We saw 20 offspring in this hypothetical cross here where B and C did separate from each other, and they were recombinant, but it's much lower likelihood than we saw with A and C or with A and B. And so, again, 1909, so it's very rudimentary, but you have to appreciate the fact that this is all happening before we have a deeper chemical understanding or structural understanding of the mechanisms behind it, that simply by looking at the statistics, they're able to start mapping these things out. Today, we can actually look at the nucleotide sequence of entire genomes of organisms to figure out exactly the location of these things. But certainly this is some pretty pioneering stuff in understanding, first off, that chromosomes are a physical thing which contain many genes. And then within that, understanding, okay, what's the relative location of these things? So that's the history and the background behind our understanding of the significance of the physical location of genes within chromosomes, and how that influences then the transmission of those genes between generations because of these recombinant events, such as crossing over, which start to mix up these chromosomes. As we've talked about before, the whole goal of crossing over, I mean, this is something that involves countless proteins that are involved in actually making it happen. It's not a accidental thing. There's a evolutionary history behind it, but the purpose of that is this diversity that it brings, as is sort of the purpose of everything in genetics, is that if you have different traits that you received from your parents, not if, you do, you have one set you got from your dad, one set you get from your mom, but someday you pass on a set to future children of yours. They're not simply going to receive all of the genes from, let's say, your dad or your mom that were on a particular chromosome. Instead, they're going to receive basically a mix that, let's say, chromosome number 12 that you pass on to a child is not going to be chromosome 12 from your dad or from your mom. It's actually going to be chromosome 12 blended between your mom and dad because of these recombinant flips that happen, these crossing over events that can happen that mix it up. And so when we look at particular traits that exist on your chromosome 12, we may see that in your future offspring, they actually received two different traits that came from your two different parents. So again, that's the underlying goal of all this, and that's how Thomas Morgan started to expand our understanding of the location of genes on chromosomes and its significance.